All the King's Men. Olfactory ensheathing cells. Axons of the olfactory nerve are accompanied by olfactory ensheathing cells along their entire length, from the mucous membrane of the nasal cavity to the glomeruli of the olfactory bulb. These are small, spindle-shaped cells about 10 micrometers in length. Olfactory ensheathing cells are interconnected by tight junctions, which ensures the formation of a microenvironment favorable for axon growth. It was traditionally believed that olfactory ensheathing cells arise from the olfactory epithelium of the nasal cavity, which in turn develops from the olfactory placodes, that are the thickened regions of the embryonic head ectoderm. However, in recent years it has been shown that olfactory ensheathing cells, like all peripheral glia, originate from the neural crest and only then migrate to olfactory placodes. The latter invaginate with the formation of nasal pits. These pits then deepen progressively to form the nasal sacs, which will eventually invaginate further to become the future nasal cavities. During the early fifth embryonic week, the ectoderm from the olfactory placodes in the upper third of each enlarged nasal sac becomes thickened and begins to develop into the olfactory epithelium. To understand the development and functions of the olfactory ensheathing cells, it is expedient to simultaneously describe the development of olfactory receptor sensor neurons. Olfactory progenitor cells, which arise from the olfactory placodal cells, undergo mitotic divisions. Some of them develop into primordial olfactory receptor neurons, others become supporting and basal cells. Initially, the progenitor cells reside in the apical layer of the olfactory epithelium near the surface of the nasal cavity. They are termed apical progenitor cells. Most of them give rise to the basal progenitor cells, globose or round cells, and horizontal cells. Globose cells are rapidly dividing stem cells that produce large numbers of immature olfactory receptor neurons. Horizontal cells are relatively quiescent, self-renewing stem cell population. They serve as a reserve to replenish the globose cells in the case of extensive damage, which can decrease the population of globose cells. Every olfactory receptor neuron produces two processes. The deep surface of each olfactory receptor neuron develops an axonal bud, axon, that will ultimately reach toward the nasal olfactory bulb. The superficial surfaces of the maturing olfactory receptor neurons extend dendritic cilia into the nasal mucosa. Olfactory ensheathing cells enclose axons with a cradle-like structure. Later, the developing axonal buds extend outward into the vascular network. Together, the developing axonal buds, the glial ensheathing cells, and the vessels form a single unit termed the migratory mass. The migratory mass penetrates the basement membrane of the olfactory epithelium. Then olfactory receptor neurons and olfactory axons grow toward the future olfactory bulb to form the olfactory nerve. Olfactory ensheathing cells always send their processes forward from the axons of the olfactory neurons, extending cytoplasmic processes around groups of axons and separating the axonal bundles into smaller fascicles. These olfactory axons segregate regionally at the early stages of olfactory nerve formation. This separation establishes distinct spatial topography, which will be maintained after the olfactory axons reach the nascent olfactory bulb. The axons from each specific receptor family are guided to a precise but different locus in the developing olfactory bulb. Thus, receptors for specific odorants are able to trace out pathways that converge on a particular portion of the olfactory bulb. The separate sites of convergence, where the axons from each type of olfactory receptor neuron come together, will become glomeruli for an appreciation of each odorant. Note that the ensheathing cells that accompany the olfactory axons arrange themselves end to end, which forms a continuous channel that encloses the axons from their origin in the olfactory mucosa to their termination in the olfactory bulb. Throughout life and the processes of olfactory receptor cell degeneration and regeneration, the tube of ensheathing cells survives undamaged, neither dividing nor migrating. Thus, the ensheathing cells provide continuous, stable, open channels along which newly generated olfactory axons can grow into the olfactory bulb and connect to the appropriate glomeruli. These unique ensheathing cells also aid in the removal of olfactory receptor cells that have died and that will be replenished by new cells. Having reached the telencephalon, the axons of the olfactory neurons, together with the olfactory ensheathing cells, merge with its outer surface, which induces the formation of the olfactory bulb. Then the axons of the olfactory neurons and olfactory ensheathing cells penetrate the olfactory bulb and form a layer of nerve fibers in it. Ensuring axon growth. As mentioned, olfactory ensheathing cells are suitable cellular substrate for axon growth in embryogenesis. This is ensured by the expression of cell adhesion molecules on their cell membrane. Moreover, they produce chemotactic and neurotrophic factors that regulate the proliferation and differentiation of olfactory neurons. Olfactory neurons live about 6 to 8 weeks. Therefore, neurogenesis constantly occurs in the mucous membrane of the olfactory region of the nasal cavity. Having reached the end of their lifespan, the neurons undergo apoptosis and are replaced by new neurons originating from progenitors in the nasal mucosa. Thus, 1 to 3% of olfactory neurons turn over daily. An injury or infection can lead to the death of the sensory neurons in the epithelium. Several studies, including investigation of neuronal turnover in the primate olfactory nervous system, have demonstrated that after injury or infection, the dead neurons are rapidly replaced via enhanced neurogenesis. The remarkable ability for continual axon growth and successful topographic targeting in the primary olfactory nervous system is attributed to the presence of the olfactory ensheathing cells that are thought to be crucial for the survival and growth of primary olfactory neurons. it has been proposed that the guidance of pioneer axons and the establishment of the olfactory nerve tracts are ultimately orchestrated by olfactory ensheathing cells. New neurons arise from neighboring stem cells. The newly formed neurons must project their axons into the olfactory bulb. Their growth is ensured by olfactory ensheathing cells, since they are, in fact, conduits through which axons grow from PNS to the CNS. Thus, olfactory ensheathing cells are crucial for the growth of axons of the olfactory neurons in both the developing and adult olfactory nervous system. Participation in synaptic transmission. In glomeruli of the olfactory bulb, olfactory ensheathing cells, together with astrocytes, surround the synapses between the axons of the olfactory neurons and dendrites of the mitral cells. This suggests that they may be involved in the regulation of synaptic function. Participation in immune responses. Olfactory ensheathing cells are involved in innate immunity reactions. They express components of the innate immune response, such as toll-like receptors 2 and 4, and can phagocytize bacteria. This plays an important role in protecting against invading microorganisms, since the olfactory nerve is the window through which bacteria and viruses can travel from the mucous membrane of the nasal cavity to the brain, providing regeneration in CNS and PNS. Recently, olfactory ensheathing cells have been considered as potential candidates for regeneration in PNS and CNS. The possibility of myelination by transplanted olfactory ensheathing cells of regenerating axons of the damaged spinal cord and sciatic nerve was shown. Thank you for attention.